Hey everybody, today we're going to be talking about Punnett squares, and we are going to be learning how to do something called a monohybrid cross, where we predict the possible offspring of two parents given a single trait. And so that is going to be our learning intention, and we're going to go through step by step how to solve these types of problems. They are a classic problem in biology and really fun to do. So you can take some time now to pause this, get the learning intention down, and I'm going to move forward. So the Punnett square is sort of the culmination of all this different terminology that we've been learning about. It uses Mendel's laws of segregation and independent assortment, dominance. We got to talk about genotypes and phenotypes. So we need to know all of these things in order to correctly use and get information out of a Punnett square. So it's a really great activity. And what we're going to be doing as we take notes today is just step by step going through how do you do a Punnett square. And again, this is something I want you to have in your notebook. A monohybrid cross is used for predicting outcomes of a single trait. So if we're doing a Punnett square for a monohybrid cross, we're only looking at one particular trait. Maybe it's flower color, maybe it's pea pod shape, maybe it's something like freckles, okay? It's a single trait. First step is we're going to construct a 2x2 grid like I have here. And I want you to write that down. Step 1, use 2x2 grid or create 2x2 grid. The next thing you need to do is you need to segregate the two parents who are contributing their alleles to this to their offspring. You just segregate these alleles along either the top side or the top or the left side. So in this case, we have mom, who is contributing either a capital A allele or a lowercase a allele, and these are going to be segregated into this row and the lowercase into this row. Dad's alleles combination would be called homozygous dominant. He has capital A, and his allele combination will be segregated across these top two columns. And again, the law of segregation tells us that we can only receive one allele from each of our parents. So mom cannot give us both of her alleles. She can only give us one or the other. Same with dad. The next step in our Punnett square, by the way, make sure you write step 2, segregate parents' alleles along the top and side. Step 3, you're going to write down, fill in the boxes to predict possible outcomes, which means that we're going to basically just distribute these different alleles now. So this allele is going to get placed into this box. This allele is also going to get placed into this box, giving us a genotype of A, or rather capital A. Similarly, this allele will be distributed here, and this allele will be distributed into this box, giving us another genotype of homozygous dominant, or capital A. Then we have the next box, so we're going to be getting the capital A allele from dad, lowercase allele from mom, giving us a genotype of capital A, lowercase a. And last, we have capital A from dad, lowercase a from mom, giving us another genotype of capital A, lowercase a. As we look at this, what this represents then is a possible offspring. So it's possible that they could create this offspring. It's possible that this would be a gene combination. It's possible for this, and it's possible for this. Okay? But it does not mean that every time mom and dad have a child, they're going to have 4 different kids with these exact phenotypes or genotypes. It is just a possibility. Okay, and now we can go through and calculate the probability of each genotype. So we can say for capital A, homozygous dominant, we have 2 out of the 4 outcomes look like that. For the heterozygous combination, we had 2 out of the 4. And for lowercase a, homozygous recessive, we had 0 out of 4. Now in order to calculate the probability, we would just turn that into a percentage. So we have a 50% chance of getting the homozygous dominant genotype and we have a 50% chance of getting the heterozygous genotype and a 0% chance of getting the homozygous recessive genotype. Okay, so now we've looked at the different genotypes that are possible that we may get if this cross were to happen. Now let's look at the phenotypes. So I haven't told you yet, but this particular allele codes for eyes in different aliens. So it's a complete dominance, meaning that if you get just one capital allele, capital A, your alien here is going to get 3 eyes. If it only has the recessive genotype, then it's going to have 1 eye. So as we look here, capital A, that's going to be a 3-eyed alien. Capital A, 3-eyed alien. Capital A, lowercase a, 3-eyed alien. Capital A, lowercase a, 3-eyed alien. All of which is to say that our phenotype is going to be 100% 3-eyed alien and 0% 1-eyed alien, because the only genotype that would give us this phenotype would be the recessive little a, the recessive homozygous genotype, which we did not get in this particular cross. Okay? So again, I'm just going to run through each of these steps so that we're all on the same page when you're doing these. First, you're going to make a 2x2 grid. Then you're going to distribute or segregate mom and dad's alleles. We'll put moms on the side, dads across the top. Remember that each parent can only give one allele, so that's what we're demonstrating here. Mom can either give us an A, capital A, or she can give us a lowercase a. Step 3, we are going to predict the possible outcomes. So each box represents a possible outcome. You're going to fill this out by distributing each of the alleles. Then we calculate the probability of the genotypes. In this case, we saw that there was a 50% chance for the homozygous dominant, 50% chance for the heterozygous, and a 0% chance for the homozygous recessive genotype. And then we go through and we say, okay, if these are our genotypes, what are our possible phenotypes? And we found out in this case that the only phenotype that we could get out of this would be a 3-eyed alien with a 0% chance of these parents giving a 1-eyed alien. All right, now what I want you to do is practice. You're going to go through and you are going to create this Punnett square in your notebook, and you're going to fill it out. You are going to cross a heterozygous green pod with a heterozygous green pod pea plant. I have it set up right here, so I've already segregated these alleles. All you need to do is go through and predict the possible outcomes right in here. And then you're going to fill in the probability of getting a capital G, the probability of getting this heterozygous combination, and lastly, what is their chance of getting this lowercase homozygous recessive genotype. Then I want you to give me the possibility for getting a green pod phenotype and the possibilities of getting a yellow pod phenotype. Go ahead and do that. I really want you to pause and take time to do that, but then I am going to post the answers so that you can double check. So take some time to do that, and then when you're done, come back and you can check that out. Okay? Okay, I hope you had time to fill out your Punnett square. Let's go through the answers then. In this upper quadrant right here, we should have had capital G, or homozygous dominant genotype. In the upper right, we should have had capital G, lowercase g, that would be the heterozygous genotype, which we also have right here. Again, that's because we're getting this capital G from dad, this lowercase g from mom. And lastly, in the final square here, we're going to get the homozygous recessive combination, which is little g. Now, probability of each of these genotypes, we're going to get capital G, 25%, heterozygous, 50%, because it's 2 out of 4, leaving 25% for the homozygous recessive. Again, that's because it's 1 out of 4. is our possibility of getting that. Which means if we look at the probability of the different phenotypes, remember we only need one capital G, one dominant allele to get the green pea pod. So we can just add these two together. This, we have a 25% of green chance of green here, 50% chance of green here. So we add those together, we're going to get 75% chance that we get a green pea pod and only a 25% chance of getting a yellow pea pod. Okay, so that is an example of how to go through and fill out a Punnett square. If you have any more questions, please let me know, and I will have a worksheet, and we'll be spending some more time in class working on this. So thank you for listening, and good luck in your crosses.